Yes, so thank you. Thank you very much for coming. Thank you very much to be at this special session of the Knowledge Summit 3 that takes place today and tomorrow, morning in France, afternoon in India. And I'm very glad to share the virtual podium with Nikhil Pal from ISI Kolkata that I will let to introduce himself if he wants now.
Hello, everyone. I think to our French colleagues and friends, bonjour, good morning, and good afternoon to our friends from India. My name is Nikhil Pal. I belong to Indian Statistical Institute, and I'm very happy to co-organize this workshop with my friend from France, Jan Bushnell. Thank you, Jan. Over to you.
Thank you, Nikhil. On my side, I'm Jan Bunel. I'm a professor at IMT Atlantic, which is a technical university in the west part of France. And I'm very proud to co-organize also this workshop with Nikhil Pal. And this workshop will take care of AI for healthcare, as everyone knows. And so just to be sure to understand what is the objectives of this workshop is that our objective is to write the Indo-French roadmap on the artificial intelligence for healthcare. The idea is that France and India has a long story of scientific collaboration, as you have heard if you came yesterday in the introduction session in the auditorium. And one of the objectives of the last Knowledge Summit 2 years ago in Lyon was to make an emphasizement, to emphasize the artificial intelligence in this scientific collaboration. So the next step for this Knowledge Summit 3 is to look back if some new collaboration have raised for the last 2 years, and then how we can imagine the future of the collaboration between India and France on the specific subject, on the specific use case of healthcare and the usage of AI for healthcare. So the idea and the objective of this workshop will be to have a very large series of presentation of different projects, different outcomes in research in this topic. And then we will have at the end of each day a couple of minutes to discuss about what should be the recommendations to propose to our government, French government and Indian government, in order to, for example, push by funding, for example, some project proposals around this topic of AI for healthcare. And so that can be about research, that can be about training and education, and for sure that can be for innovation and transfer. So this is let's say the main roadmap of this 2-day workshop. And let's just me to introduce the program of this 2 days. So the first day, day one today, we will have exactly 8 presentations, half from France and half from India, which will be scheduled as 3 or 2 presentations in a row with no question. And then each time we will have 10 to 20 minutes of Q&A session for the previous speaker. And then at the end of the session, we will have a general discussion with the panelists. So all the people that have taken the microphone this morning in order to elaborate a perspective and conclusion. So just a remark today, we will have a switch with two speakers that for unfortunately schedule constraints. So Dr. Pritesh Sethi was supposed to start, and finally it will be Shanda Ahmad that will start, and that switch with the talk of Pritesh Sethi. So that's why it will be quite different of the agenda that you have in the website, because the website cannot have been updated. And for the day 2, We will have also a series of talks, half French and half Indian, as today, with scheduled with several Q&A session also, and we will have a larger closing session, closing talks and recommendation and by panelists in order to write the conclusion of this workshop. So I hope I have been clear, and I hope we will have some fruitful discussion, fruitful recommendation that we can send to our government at the end of this today. And I propose, as it is exactly 10:10, so we are exactly on time, to give the microphone to Chandra Hamad, which is professor in the School of Computational and Integrative Science at the Jawaharlal Nehru University in Delhi. So I will stop to share my screen and give you.
Thank you very much, Jan. And bonjour, good morning, and good afternoon. And first of all, my apologies for disturbing the schedule and requesting for this rescheduling, swapping my talk as I was summoned for something at the previously scheduled time. Thank you for accommodating that. I'll straight away share my screen. Is it visible now?
Yes, it's perfect. Thank you.
Okay.
So I will ask our speaker. As you know, you have 15 minutes, but it's including the technical change between speakers. So if you can leave in 13 minutes, it will be perfect. Thank you.
Okay, thank you. Just I'll check my slides are moving just to make sure. Is it? Can you see my slides moving? Sometimes the screen is shared without the slides getting moved. Okay, so thank you for that. So today, without wasting time on my additional apologies that I need, I'll start my talk, multi-scale and multi-dimensional modeling of biological and medical data. Primarily, I'll advertise my own lab and tell you about what we have been doing in our lab, what have been our success or efforts that we have been placing in. But I quickly take the luxury of starting with a poem to put it in perspective. Physics is fundamentalist and fussy. I came from physics background, so I can take the liberty to say this. Mathematics is dumb and yet not easy. Astrology is hasty and crazy. Human brain uses none, for deep inside is terrain. It knows to represent, test, and train without bothering to explain. Artificial rules this world, for even if a solution is transient, predictions noisy and incoherent, it works the best for most because it is, after all, intelligent. So this is the perspective. So I'll quickly go through three aspects of my presentation. In the first, I'll mention the ongoing CEFIPRA funded project, what we have been doing here. Then I'll tell you a little bit about ongoing and recently completed projects where we used AI to solve some interesting problems, and the key achievements, slightly older story, which I'll touch upon based on the time I've allowed. I'll just touch on those things which we can do. So our CEFIPRA funded project is with Pedro Ballester in Maxwell Cancer Institute. And this is to do an interpretable AI for oncology. The primary goal is to find certain features or find the AI-based models so that we can predict the drug prognosis, whether a particular drug will be effective for a patient or not based on the profiling. So it is, in that sense, it is a personalized medicine project. In a broader perspective, in the narrower perspective, we have the PDX data, and from the multiple sources of data, multi-omics data, which includes the transcriptomic data, CNV data, etc., as have been shown in this first panel of the slide, CNV, CNA, SNV, gene expression, etc. We pull features, can we represent them in a lower dimension and try to develop model, because the amount of data we have or the number of patients from which we get the data is much smaller than the number of dimensions in which we are dealing. So these are the results so far. We have also tried to integrate whether can we have a pan-cancer model, so to represent all the features and train our AI and deep learning models based on the pan-cancer reduced representation. This is the project that we are doing. And the second, other than the CEFIPRA funded projects, we have been also working on this drug discovery project, CMAP. And there we have integrated the gene expression omnibus datasets with the CMAP datasets. And the main idea is, can we in all the expression profiles that are deposited in the NCBI in gene expression GEO, can we integrate them, provide a uniform platform, and where in some of these platforms, microarray platforms, for example, many of these genes are absent, can we now complete these expression profiles for the absent genes and create a holistic universal matrix for or global expression matrix? That is what we are doing, and we are using deep learning models for them. And the model for kind of extrapolating or expanding the large scale missing values in these expression profiles, we are using a DGX model for now. One more project we did recently was because COVID, everybody was working on COVID, and it is a kind of Me Too activity. So we contributed by trying to look at the age distribution profiles of different patient groups. And initially, when the COVID patients also had a travel history, and we could actually trace every individual to where this strain, the COVID strain, could have originated, and looking at the data from these travel histories and the expression profiles, the age profiles of a population group in different states, we were able to find some correlations between the age profiles, the mortality in male versus female, etc. And this work we published recently. And this is what we are doing. We used simple AI, classical machine learning, for that. One thing we also did was to participate in the competition to predict the single cells which associate to a specific topology in Drosophila in this competition dream. And there, I just skipped to this. We were not performing on the top 5, but we were in a couple of these competitions at 7, 8, and 9. So another project we are doing is to predict modeling the ECG signals, and from the ECG signals we are trying to we are trying to predict by taking, say, 1 hour or 2 hours ECG signal data. Can we estimate for specific locations where a particular anomaly has happened. So this is useful, for example, for interpreting and to extract useful regions from Holter collected data, etc. And this work we also published recently in Computers in Biology and Medicine. And we went one step ahead. And instead of just predicting that there's an anomaly, we could also predict 7 different types of anomaly with some moderate level of success. So this is what we did. And then we also worked in collaboration with Padova University with the group. And in this, we were trying to look at what is the optimum data set that is useful, that is required for making a deep learning model. And at certain data size, the deep models perform better. But before that data size, actually the classical machine learning models such as SVM implemented on PCA, etc., could actually be more effective. So we looked at the data size requirement for individual cases, for a specific case, MRI, where we were trying to predict the cognitive performances, the linguistic performances to be specific. In the third aspect that I mentioned in my introduction is we have been working for a long time on the protein-DNA interactions, the gene expression and how they contribute, looking at the proteins which bind to DNA, looking at the DNAs which are suitable for binding to a transcription factor, etc. And in this, we have used a deep learning more recently to predict the DNA binding proteins from sequence. And one problem that we solved was that the sequences are of different sizes. If we convert them to evolutionary profile PSSM, they are of different sizes. So can we use fixed size image based predictors such as CNN to predict the DNA binding proteins? And then we developed certain padding schemes and we made them equal sized images. And this method worked quite effectively. This was published in Proteins recently. So this is the older one.
Excuse me, Dr. Yeah, just are you still in the first slide, because the slide is not changing in our.
That's what I asked. My slide is changing or not? No, I'm already on my 19th slide.
19. Oh my God.
Yeah. I'm so sorry about it. So that's why sometimes slides don't change. That's why I asked you in the beginning, is the slide changing?
Yes. I heard that you asked if you are able to see your slides.
So now, so when I go to the presentation mode, that sometimes the slide disappears because it is sharing a different screen. That's why, yeah. So
now, please continue without the slideshow mode, and so it's perfect now.
Okay. So anyway, I'll share these slides with you later on if anybody's interested.
Thank you very much.
Okay. So. But I'll quickly browse through the slides so you can have a look at what I showed you. This is what I showed about the Cephipra project. This is the model. Without the slides, actually, what I'm saying makes no sense, actually. So this is the gene expression omnibus, DGX model I talked about. This is what I talked about, the age distribution profiles in different genders and from different infection origins, Middle East, European Union, UK, etc. We defined different lineages based on the travel histories. And this is what I showed you in terms of the DREAM project, where topology predictions were carried out. This is where our team is stood. And this is what I was talking about, the ECG signals that we take the ECG signal, we make two-stage model of the deep LSTM followed by an SVM so that the normal ECG can be distinguished between anomalous one, and the anomaly could actually be, a specific anomaly could be predicted here. So this is what I showed you earlier, and this is the paper that I was talking about in terms of the MRI images and predicting the lesions from them. So then I was talking about in the slightly before that, we have been working extensively on the genomics and proteomics aspect, large scale throughput, high throughput predictions of DNA binding and protein DNA interactions in different aspects. One is what kind of proteins bind to DNA. One is where the protein will bind. Why, and the distinction between the sequence-based motifs and the shape-based motifs, and why some of the motifs in the genome actually bind to a transcription factor while others do not, and what are the cofactor principles, etc. These are the issues we have been dealing earlier. So in most recently, what we have done is to look at the evolutionary profiles of proteins, and from the evolutionary profiles, can we predict that this profile comes from a DNA binding protein? And what we developed here was a technique to pad to make all the PSSMs of the same size. So kind of different padding schemes. What is the most appropriate padding scheme for this? Prior to that, we were working on the exact binding site on a protein where the protein will interact with the DNA. And for that, we developed a number of web servers, quite widely used ones. We published several papers in bioinformatics, BMC journals, and nucleic acid research. And these web servers provide basically AI-based methods of interpreting a sequence and finding specific regions on the sequence where the DNA is likely to bind. So we examine the conserved residues, again with the help of the clustering principle, unsupervised methods of machine learning. And in some cases, the supervised one where we know about the free energy changes that are caused by removing a residue from a protein. And this method and statistical principles of the protein DNA interactions could be utilized to develop what is called the knowledge-based force field. And we developed this method called readout, which can actually take the protein DNA complex structures and identify novel sequences which will fit into the complex. Effectively, this was used for predicting the non-canonical binding sites in SAA gene, experimentally confirmed. So I would rush quickly. One issue that we were dealing with also about the cellular specificity of transcription factor binding sites, where actually one transcription factor binding in different cells, very few sites are overlapping with one another. So we introduced the concept that we can have a sequence regions and sequence all the possible tetramers. We carried out a molecular dynamics simulation for them. And these simulations were then converted into ensembles. And therefore, now we have a dictionary of the tetramers or longer flanking regions, maybe pentamers, and how these pentamers will have what kind of a conformational ensembles they have. This conformational ensemble can be used as a feature of the entire genome, and therefore we can make predictors for the genome-wide behavior of transcription factor binding sites. And we were able to identify or detect interesting things such as the modular regions around the motif, the DNA transcription factor binding site. It's not just the motif which is important, but also the surrounding regions. And the shape of the surrounding regions can actually be used to characterize them and group them, cluster them into useful patterns. So most recently, we are also working in collaboration with the Manchester University, Nicoletta Bobola, where we are looking at Hox transcription factor binding sites in branchial development, HOX2 and HOX3. They have specific binding sites, but not much is known about why a particular transcription factor is selected by certain sites and by not others. So these are we have some interesting results which may be shared on a different occasion. I will wind up here as per the mandate of my talk. This is the lab members that are working with me. Thank you.
Thank you. Thank you very much for this large contribution and a lot of project and publication. So thank you very much. It's impressive.
Thank you.
So please stay with us for the Q&A session that will take place in 30 minutes. So now it's my pleasure to introduce Nicolas Farrugia. So he is actually associate professor at IMT Atlantic and researcher in the Labstick research laboratory in Brest, so which in the western part of France. And so thanks, Nicolas, to be there, and the microphone is yours.
Thank you very much, Jan, for the introduction. And I'm really, really honored to be presenting this work today. So thanks for Shanda for his awesome contributions from the last talk. I mean, what I'm going to present here is much more narrow in a sense, because we're a very young team and we are very honored to be able to have the opportunity to talk today. So as Jan said, I'm part of EMT Atlantic and also part of the Department of Mathematical and Electrical Engineering. And today I will talk about better representations for artificial intelligence, which is also the name of my team that I'm running together with other colleagues. So in a nutshell, to give you a small outlook of what I'm going to talk today about, we want to know, why do we need better representations for artificial intelligence? So this is the main question. So in order to explain that, I'm going to recapitulate successes and limitations of recent AI approaches. What I mean here is mostly like machine learning and of course deep learning. And then I'm going to showcase a few results from the brain team and some applications to healthcare engineering. So first, we're going to talk mostly about machine learning and deep learning here. So what you have to understand here as machine learning is really like the paradigm to infer like a complex function from inputs and outputs in a supervised way. So that's the simple paradigm. The difficulties posed by this is that it's a very ill-posed problem. There's like an infinity of potential solutions. And in traditional machine learning, the main approach is to seek for particular solutions. The revolution in deep learning is that we can actually find specific solutions by combining three interesting elements. The first element is a compositional approach. So to express the potential solutions as actually an assembly of small functions, as atomic functions that are usually called layers. The second ingredient, so to say, is end-to-end learning, which means tuning all these atomic functions together. And the third ingredient is differentiable differentiable training, is to optimize the functions using variants of stochastic gradient descent. The ambition of this kind of work, of deep learning, is to become the new informatics, so to say, like the new way to do science in a way, applied to many, many different things. So as most of you know, deep learning has successes in many, many different fields, in computer vision, in protein folding, finding in biology, in speech recognition, in aiding programming, computer games, speech processing, and of course, health. But there are also many limitations. So I'm going to introduce a few limitations which are known in deep learning and explain what we are trying to do to address this limitation. So the first limitation is computations. So I'm going to mention here, but not going to detail much more. That's one of the part of the work that I will not have time to detail here. So deep learning is very efficient, but it's very compute hungry. So this image comes from OpenAI, and it gives an idea of how much the need in computation has grown with the state of the art in any domain, in language, in vision, in speech, in games. So there's a lot of effort which is given to compressing deep learning architectures. It's one of the things we're working on. One of the other limitations is data, and especially the lack of data in certain cases. So this picture is from Josh Tannenbaum, and it's here to illustrate that actually humans are able to do few-shot learning, which means learning from few examples. So if you see in this picture, the three examples highlighted in blue, that's objects that don't exist, that are virtual objects. And if I ask you what are the other objects of the same type in this image, it's very easy for a human to do it. Actually, even like a child can do it. But if used in the traditional deep learning approach with no special effort, it's a very difficult task. So that's one of the other limitations. In certain scenarios, like actually in healthcare, we don't have much data in certain cases. We have like a very limited set of annotated data. the second limitation. And the third limitation is that we do have data with a very complex structure in certain cases. So on the top left of the image, I represented a convolutional filter, which is what is used in convolutional neural networks. So in 2D convolutional neural networks in computer vision. And we are able to do this because images are organized in grids of pixels. So 2D grids of pixels. So an image is a matrix of pixels with different values, with RGB values. And we're able to exploit the neighborhood between pixels to define a convolutional filter, which can share the weights between neighboring pixels and can also do the same processing at all the different parts of the image. And this is possible because an image is a grid and has a very regular structure. So the thing is that there are other things that images. On the right side, represented like a series of very complex one-dimensional signals coming from different waves, and that can be found in complex problems in physics or in audio processing, for example. And in this case, the filters that would be needed to process this kind of data are much less regular. We have problems of nonlinearity and non-stationarity. On the bottom, I represented a graph, which can be, for example, a social network graph. And this kind of structure is a very high dimensional object, and the features have relationships. So the different features have relationships, but the relationships are not regular. They're not a grid. They're not like a 2D grid, like an image. And that would also be the case for example, neuroimaging, brain imaging, in which we would have regions of the brain connected through white matter fibers, for example. That's not a regular network. So we need to think about methods to be able to deal with these complex structures. So in the brain team, so better representations for artificial intelligence, we are 3 professors, so to be more accurate, 2 associate professors and 1 research director, 2 postdocs and 12 PhD students with collaborations with many different institutes and in the public and the private sector. And we're working on 3 aspects of methodology for artificial intelligence. So the first aspect we're working on is the compression of deep neural networks. So I mentioned it in the beginning, like the objective is to be able to reduce the computations and the memory requirements of deep networks, because deep networks have become very large. The second main pillar we're working on is graph signal processing for artificial intelligence. So that tries to answer, attempts to answer the problem about structure in data. The fact that data, lots of fields have high dimensional data with complex structure that are not regular grids like images. And the third methodological aspect we're working on is few-shot learning. So that's for the methods. So Brain is primarily a methodological AI research team, and we have 3 main application domains. So some of us are more focused on trying to start from the application and nourish the developments and in the methodological fields I mentioned. And the 3 main fields we're working on today are computer vision, mostly like classical benchmarks in computer vision, audio processing, so really like processing sound, and brain imaging. So for today, I will mostly talk about the part on the right side, which is graphs, neuroimaging, and few-shot learning. So regarding few-shot learning, we had very recent successes in this field, as the Brain team is ranked first in all the classical benchmarks in few-shot image classification. So you can check that on papers with code. So that's from like datasets like Cypher few-shot, Mini ImageNet, and Tiered ImageNet. And that's from the paper from Yuqin Hu from 2021. And this is something that's been we've been able to push the state of the art by actually exploiting the data structure in using graphs and with ideas from transfer learning. So that's a very general image classification task, not specific to healthcare engineering. So in the rest, I will mention more applications related to brain imaging. We've tried to apply the same few shot techniques to brain imaging in the work from Myriam Bontunou. So the idea that Myriam did, so I'm not going to give much details here, but we've developed a benchmark for few shot brain decoding. So few shot learning of brain activity. So really trying to decode mental state from cognitive tasks using functional MRI. And we used the IBC dataset, so that's the individual brain charting dataset from INRIA, from Paris. And we've showed that we could actually use some of the methods that are used in image classification in training few shot brain decoders from scratch using.
Nicolas, be careful, you have mute. Just for the last sentence, you have been muted.
Okay, sorry, I don't know how that happened. Sorry, I'm gonna have to go back a bit. Okay, so I can start from here again, I think.
And if you can just resume the Zoom a bit, because we don't have the bottom of your slides.
Really?
Yeah, just it's not a big deal from the beginning, but if you can reduce a bit by your Zoom back, it's a bit better. Oh, perfect.
Okay, so I guess I just present like this. Okay, sorry about that. Okay, so I guess I was finishing by saying that we developed a few shot learning benchmark on brain activity decoding using the IBC dataset. So that's something you can check out in recent publications, but essentially we were able to adapt the few shot benchmark from image classification to neuroimaging. And that opens new possibilities because we can imagine that in certain situations, we don't have many examples of a very rare brain disease or for biomarkers. So in the last part of my talk, I will try to summarize the use of graphs for neuroimaging, and in particular the use of graph signal processing. So in the recent years, the rise of network neuroscience has shown that we can actually model the brain using a graph by defining nodes as brain regions and edges as different ways that can connect brain regions. For example, diffusion imaging can model the white matter tracts. So we can use tractography to build a brain graph, but we can also use what is called functional connectivity, which is a correlation between activity signals. So once we have a graph, we can model the graph using graph theory, which means having an adjacency matrix, and then find different descriptors of this graph, like communities and hubs and paths. But we can also use graph spectral theory, which is a way to actually decompose the matrix that represents the graph and derive what would be called in the literature gradients. A new framework has emerged in the last 10 years, which is called graph signal processing, which is actually to generalize all the classical signal processing pipeline to irregular domains on graphs. And we advocate in this paper that we can actually interpret signal brain activity on gradients by using gradients as a Fourier basis of brain activity. So low frequencies and high frequencies can be seen as like more coupled or less coupled to brain structure. So we've applied this kind of framework to different applications. So in 3 different papers, we've shown that we can actually combine this kind of graph signal processing with supervised learning and feature selection. And we show that we can actually boost classification by informing supervised learning like AI using the graph. So graphs can be used as feature extractors in order to do efficient learning with very few features. Another type of application is to define graph convolutional networks using the graph for new convolution operators. So this is done this was proposed in a few papers and done also in collaboration with Pierre Bellec, in which we propose a framework for brain decoding using graphs to define a graph filter. So that's 4D task fMRI data, so temporal data, that is decoded on a brain atlas. That is actually parcellated on a brain atlas, and then the brain graph is used to define special graph convolutions, which are called Chebyshev networks. And then it's done using, it goes through different layers of graph convolutions and then the MLP for final decoding. And this has like really good performances for decoding on human connectome project tasks. So I'm just going to give quickly a few perspectives. So the work I've presented here will be applied in the future in like projects that start now for new projects in brain-computer interfaces. So we face like very difficult problems in this kind of applications due to very low SNR. So we're going to try to translate all the things we've done in functional MRI in electroencephalography, which is much more noisy, with a lot of variability also between sessions and users. So the goal is to really to build on the few-shot learning and transfer learning to generalize that to brain-computer interfaces. And to finish, another application that is just starting now is also a health application. It's a collaboration with a company that actually does sound event detection in bedrooms in elderly houses. And we plan to do AI-assisted diagnosis and detection of rare events. So like anomaly detection. And this is a tool that is used to help the medical staff and to have like monitoring during the night so that to better organize the work for the medical staff. And this is also, we are also facing like very difficult questions in viability in the data and annotations that are costly. So we plan to apply new things like self-supervised learning for this work. Thank you very much.
Thank you very much, Nicolas. Just in time. So again, I recall that we will have the Q&A session just after the last talk of this first series. And so now it's my pleasure to introduce Dipti Prasad Mukherjee, which is a professor in computer and communication science at the Indian Statistical Institute in Calcutta. So thank you again, Nicolas, for this very valuable presentation, and I'm very happy and glad to have Professor Mukherjee to the next presentation.
Oh, thank you, Jan. I think I'm audible.
Yes, it's perfect. We can see you.
And I'm not going to take the risk of going to the PPT in a display mode, so I'll just go on doing this.
Perfect.
Good afternoon and good morning to all of you from France, and I'm really thankful to invite me to give a talk. I'll be talking on a very recent work that we have started, so it's kind of work in progress, and that's exploring non-invasive tool for monitoring cancer biology. But before that, let me just introduce some of the recent works that we have done. This is our institute, Indian Statistical Institute, as you know, was founded by Professor P.C. Mahalanobis, and probably at some point of time in our career, we must have used this Mahalanobis distance. And this is just to introduce my institute from Calcutta, actually, here. Okay, so these are the three specific applications I'll be talking before I go to the topic of non-invasive prediction. One is the shape analysis based, then there is a device specific application and an image and texture based application. These are my co-authors working with me from University of Osaka, and what we did is the disease specific organ shape components modeling, and I will explain these in next few slides. So these are 3D shapes of liver. And as you can see from the variations of colors, that these are modeling of the global shape deformations. But the challenge really in this aspect is, can we find out the local shape deformations? So can we find out shape components, or if you think of some kind of shape vectors that can locally identify the deformations? Let me give you another specific examples with femur. You see this femur as it progresses over time, and you can notice the deformations in the femoral parts. The question is, can we identify those deformation regions and mark it, and so that a proper synthetic femur when designed, that kind of deformation can be taken care of. There was an interesting paper as old in 2003 on in SEMC graph actually, where human body shapes were modeled. So there is a statistical shape model, and by changing the different parameters of this shape model, you can actually generate different 3D shapes from one single data set. Question is, can we do that for this case? And what we did is we built a statistical shape model of the organ, in this case specifically on the liver, and then we actually gave a model of use of orthogonal complement of this shape model. So you can assume that there is a healthy subspace which represents the healthy 3D organ of, in this case, the liver specifically, and then all the disease components, we actually sort of try to show that those disease components are orthogonal complement of those healthy subspace. Here is another example that we worked recently presented in ICAS. It's a segmentation of ultrasound images, but specifically for phlebotomy applications. Those who are like me who do not know what is phlebotomy application, it is to have a small ultrasonic devices, and you put it on some of the body parts, and you would like to actually visualize the veins and arteries under the skin through a small display on this device. So the question is, once these veins and arteries are scanned, would it be possible to reconstruct these tubular sections representing veins or arteries? So as the ultrasound, handheld ultrasound devices are placed, you can get C scan, and you can also vertical scan or the B scans of those tubes and or arteries or veins. And in our case, so this is an example of the vertical scan or the B scan of those ultrasonic devices. These are examples of C scans, and in the C scans, the challenge is to identify that vessel inside, which dives, which turns, and since it's a handheld device, there are umpteen number of kinds of noises. So question is, can we clean those noises, and can we develop a filter by which these tubular sections can be extracted? So we have used a specific speckle removal model, and through the repetitive filtering process, which includes the edge fidelity term, how good we could maintain the edge even when we are doing a diffusion kind filter. So it's kind of an anisotropic filter that I have used, and with these such constraints, we have generated these segmented results on the original C-scan images of the handheld ultrasonic devices. Once these sectional images are once the sectional images are created, then we could do the 3D reconstructions of that. All the slices are placed one after the other, and then these 3D reconstructed volume in the form of those vein structures can be generated from the segmented results. The last work that I'm going to present here is actually developing a better random forest techniques and for detecting mitosis. The problem is well known. It's just to separate the mitotic and non-mitotic cells. Unfortunately, the challenges in identifying mitotic cells is that there are, or separating mitotic from the non-mitotic is, there are different other shapes that are present, and for example, these external object and then dense non-mitotic objects, lymphocytes. So separate them out and identify the mitotic cells. Even mitotic cells has got different phases, and that gives a difficulty in identifying these prophase, metaphase, anaphase, telophase, etc. So with our improved random forest techniques, we could achieve better classification results, and the area under the curve compared to the competitive approaches are are better. That's what we've seen, that we have shown here. Now coming back to our my presentation, that we are trying to explore the non-invasive tools for monitoring cancer biology. We are actually collaborating with a reputed cancer center in Kolkata, which is the Tata Medical Center. And what we are getting from them is the PET-CT images. And so here is a typical visualizations of the PET-CT image, where we can actually see the tumor growth. And the idea here is to actually replace the non-invasive, so replace the invasive needle biopsy operations and relate. Excuse me. Okay, so the objective is to look at this growth of the tumor and model the growth of the tumor with respect to the different histopathological parameters that we get for the patient-specific informations. So what we get is PET images, we get the CT images, we get the fused PET-CT images, and then a set of histopathological parameters for those patients for whom we have already the PET-CT images. So typically this is the histopathological parameters that we get, and that includes these gender informations, these ER and PR values, estrogen receptors and progesterone receptors. I will come back to that, these items once again. And then we have these data for human epidermal growth factor receptors, HER2, age information, tumor grading information, breast cancer. So fairly a good number of good modalities of information, histopathological data are available for the PET-CT images. And presently we are working with 123 such data sets. So what essentially we are trying to look at is to model this table. This is for the LABC cancers, locally advancing breast cancers. There are 4 such types of LABC: luminal A, luminal B, HER2 enriched LABC, and then triple negative type. And these estrogen receptors, progesterone receptors, or HER2 and the other clinical grade informations are available for different subtypes. And the question is, can we model these different subtypes of LABC corresponding to these histopathological parameters, so that we can come up with the information of the grade or stage of the cancer, or even can we predict ER and PR parameters from the PET-CT images? And the objective is, of course, to replace the needle biopsy and to reduce that time that it takes. First of all, it's a painful process. Second, it's time consuming. Even at least 2 to 3 days are minimum required to have a reliable invasive needle biopsy result. So such a modeling should actually open up a lot of other possibilities. Okay. So therefore it's clear that our objective is to get some textural image features of the PET-CT images, and then model that relationship between these textured features and textured features of the PET-CT images with the histopathological parameters. We would also like to do a 3D reconstruction of those tumors, and we would also like to add a treatment response variable, which is a number between 0 to 1, but provided we have a sort of a temporal data of disease progression. Now, currently we do not have that temporal data. The doctors are in working with that. So it'd be interesting to look at these treatment response variables for such cases. So these are some of the standard features that we have worked with, and then we have actually, so this is kind of the pipeline that we worked with. We have those PET-CT slices, we fuse them to have a 3D reconstructions, develop the 3D features, and then either go for the binary classifications, considering each of these subtypes. Remember, there are 4 subtypes. So what is the response of luminal A versus luminal B, or the subtype HEART2 versus luminal A? So these analyses we are currently doing. These are some of the results that we have of 3D reconstruction. Unfortunately, because of the number of availability of less number of slices and more of interpolation between the slices, the result at present is not very smooth for 3D reconstructions, and we are looking at that. We are looking at an exhaustive set of radiomics features, first order features, shape features, gray level co-occurrence, and the zonal matrix features. We also working with these gray level dependence features between the neighborhoods of the tumor and the adjoining areas. So one of the specific features that we have introduced, and we have seen good results coming out of it, is the isocontour features. So given a tumor biology, tumor shape, we would like to slice them into different intensity levels and look at the contour of those slices. And then we have actually introduced a sort of a winner contour features. So some contours we would like to remain, other contours we would like to throw away, and we have introduced certain criteria for selecting such features. Okay, so I'll just skip. These are some of the initial results that we got. We have some feature selection techniques. Unfortunately, the results are not up to the desired level. So again, once again, the model is directly from the PET or the PET-CT fused image. If we could predict the grade of the LABC. These are the area under the curve measures. These are the variations in predicting. Then we have results for what is the effect of predicting the estrogen receptors or progesterone receptors from the textures itself. That would be, if you could do it, and this could be a very good work in the sense that you won't need the needle biopsy for such image-based predictions. Okay, so these are some other results where we are actually comparing between the 2 subtypes of these locally advancing breast cancers, and kind of doing a binary classification given a set of features and given a set of histopathological parameters. So overall, the project has different challenges. The main challenge is, of course, feature selection, and we are not yet there. We are actually currently exploring some deep learning techniques to have a better feature selection. Even though that area is not that much explored, I must say. The challenge in modeling the histopathological parameters and image features, currently we are using logistic regression-based techniques. We need to explore that once again. We need to get temporal data for disease progression, and that's going to give us a very good idea about how the disease progresses and how good we can predict that. And finally, a smooth 3D reconstruction to get a good results. Thank you. Thank you very much.
Thank you. Thank you very much for this valuable presentation just in time. So now we have time for Q&A session. So if the Thuda speaker can just turn on the camera, and so we leave the floor to the audience. If you want to ask questions to our speakers, do not hesitate. Please just unmute your microphone and start the question session.
We are so our result for the time being, for example, this non-invasive type modeling is not what we are expecting. I think we need to go a little bit away from these logistic regression models that we are currently using. But before that, I think one of the key challenges that we are facing is the feature selection problem. And because this is a well-beaten track and people have worked with texture analysis for I don't know, maybe 30, 40 years, and there are hundreds of features. The question is what kind of features we are looking at. And PET images, particularly the PET CT that we use, doesn't give good texture information. Then what else? So is this shape feature important? To reconstruct the shape, we probably do not have enough data for 3D reconstruction. So such challenges, and really we are not sure at the moment that that the kind of stress should be on the feature selection area or the classification. My personal take is going for feature selection in a little bit bigger way, and instead of using the horsepower of deep learning, we use the techniques of deep learning to come up with better feature selection. So that's probably one way to improve our results.
So I have a question, Dr. Deepti. You didn't use the horsepower of deep learning. What is that specifically that you've done? Second question is, how are there any startups challenged with you or working with you, or is there a room for startups to work with you? And of course, the third and the most critical is you set some 135 set of data or something.
123, yes.
Right. What is that we can do together to have probably 100 times the data?
100 times, I'm not sure. I mean, well, the any cancer center having a PET CT facility can generate these data, but these are of course the de-identified data, so de-identified patient data. But certainly we need data. But in the medical imaging, it is always a challenge. It is always a challenge that you do not get enough amount of data. But what we are lacking in particular is the time progression data. And in India, I think it's specifically very difficult to, the doctors are so busy, particularly the radiologists. So it needs monitoring of that particular patient over a period of time, maybe even 9 months or 1 year. And for specific therapy, how it changes, how the tumor progression changes. So yes, so that is a big challenge. Right now, there is no startup that we are really.
Well, that is what we thought is if we can put a few startups in the arena around the doctor, because we know that doctors will never be free to do this kind of activity.
Exactly.
So we need to support the doctors through the startups who will benefit out of this, and the doctors will also benefit, and then see how we can have this progression recorded over a period of time and which would be useful.
Exactly, because it's a non-invasive. It's a non-invasive tool.
Absolutely.
So
it's something which is.
We want to replace the needle biopsy. We want to replace the needle biopsy. So that's a big challenge, big dream.
So that's a very wonderful work that you're doing. It's just that we need to move on further and see how we can collaborate. And I would love to interact with you after.
Okay. Thank you. Thank you very much.
Thank you. If there's another question from the floor. Okay, so maybe I can ask from Nikhil, maybe you have a question?
Yeah, well, since no one is asking, so my question is addressed to Dr. Nicholas. So if I understood that when you talked about few-shot learning in the context of brain images, when you say few-shot, I mean, what is the number of samples that you are referring to? The reason I am asking this question because you followed this up with finding of biomarkers from this few-shot learning. So if the number of samples are really very small, I mean, how reliable would those biomarkers be? Could they be even outliers?
Okay, thank you for this very good question. So few-shot learning applied to neuroimaging it's a relatively, well, I mean, it's a relatively new field if you take it from the deep learning angle, but people have been showing that you can actually learn with few examples for quite some time now. What I mean by few shot here would be, so we've tested 1-shot and 5-shot scenarios, so 1 to 5 examples, but that's just at generalization stage when you introduce actually new problems new classes. But to be able to do that, one of the usual way to do that is to first have a generic data set, which is a data set of a large size. So the kind of questions you can ask are, you have like a large, let's say in a healthcare context, like you have a large data set of healthy subjects in which you're going to try to do a benchmark task like age prediction, let's say on a large set of subjects. And that could be done using a deep learning backbone, for instance. And once this backbone is trained, there is a variety of techniques, a variety of ways to do few-shot learning to generalize to new data. So this is actually a kind of a generalization of what is known as transfer learning already. But so once you have the generic data set, train the backbone, you can reuse part of this backbone and do 5-shot classification for example. That's the benchmarks. The benchmarks already show that you can do such things, but we've not done it in a biomarker, like in a disease classification type of benchmark. This has not been tried yet.
Thank you. Brain images has an extra advantage because you have some kind of quote unquote model for the brain. So maybe you are muted.
No, what do you mean by model of the brain?
Well, you have some idea about the structure of the brain.
Yeah.
So if something unusual happens, yes. So that is extra addition at some advantage compared to other areas where we try to do few-shot learning.
So we've tried to do this. We've tried to incorporate knowledge of the brain structure, especially knowledge of connections, of like brain connectivity, in order to better inform the backbone training. And so far, we didn't see a clear benefit in using brain connectivity, but we're still working on it. But indeed, one of the things that make few-shot learning possible is exploiting the relationships between the features and also between the samples using models, using graph models. So this is a very relevant avenue for future work indeed.
Thank you.
Thank you. Thank you very much. If there is some other question. So maybe I have a difficult question. Maybe we can start with Professor Mukherjee and then Professor Farrugia for the last question for this Q&A. If you want to think about perspectives and about what is the most key challenge in your field for the next, let's say, 5 years, what should be your imagine that you have you can have full funding about something. What should be your direction of research and the next step that should be done?
Yeah, I mean, given a problem, I would stress feature selection, and then the feature selection using these deep learning or the CNN architectures, because most of the applications that we are looking at is essentially doing a classification. It's a very general, broad term that I'm trying to use, but I really do not seek some kind of engineered feature selection given a problem. So given a problem, engineered feature selection using these deep learning architecture is what I think is an area where I think at least I would love to concentrate in coming years.
Thank you. And for you, Dr. Farrugia?
Well, I would be really happy if we were able to show that deep learning can actually be done in much more accessible context. What I mean by that is that this kind of technology is already very costly in many aspects, costly in terms of resources for training, but also resources for using them. And I mean, it's nice to do academic research with large data sets and have like computing power available in clouds, but it would be great to be able to reduce the barrier for the availability of such technologies for the general public and for medical instances, for medical like medical structures, for maybe, you know, like small associations, citizens. And I think there is a lot of research to be done to be able to reach out and to be able to communicate, to not just communicate, but reduce the entry barrier that is needed for this technology. So that can be called something like low tech kind of AI, being able to do like this kind of approaches with much less resources and much less budget. And it's not just a matter of developing efficient learning hardware or things. It's also theoretical questions related to questions we were trying to address through compression of deep neural networks and few-shot learning.
Thank you. Thank you very much to both of you. I think unless there is some last question from the audience, but I don't see anyone who is
Jan, may I have the last one?
Sure, sure.
Hello.
Sure.
Yeah, this is actually to all the speakers really. So deep learning, in my view, it's fantastic in terms of results, but for medical application, as of date, it's deep black box. I mean, I think that's pretty dangerous if I want to use it for medical applications particularly. I do not know how Professor Mukherjee and Professor Nicholas would react to this.
Yeah, it's a good point, and I think it's a debatable point. But yeah, but I do not see any problem with that. I mean, because even doctors are not perfect, so, and even you cannot always rationally explain a doctor's decision. So why put all the blames on an architecture that you demand in proper explanation? So so that one of the take, but yes, I understand that there are problems and challenges. Yeah, but rather I would agree with what Nicola was stressing on, that we have to have some lightweight, easily accessible, consumer-friendly applications in these areas, and probably deep learning may not, or some other versions, some compressed, smaller versions of deep learning might help in that respect. I mean, that's just my take.
Well, I'm pretty much going to agree with Professor Mukherjee here. I mean, yeah, somehow doctors can also be a black box in themselves. But if you take it from the citizen's perspective, from the patient perspective, actually, you sometimes don't really have, or you also don't have to have the elements to understand a doctor's decision or doctor's diagnosis. And that's kind of fine. I mean, things are working most of the time, let's say, when you're facing, like you have a problem and you ask a doctor what's going on. But of course, explainability is also a research field. So that's something that can be addressed. It's difficult to address it if you have very, very, very large models with lots of non-linearities. So the best explainable models are the linear models, and we can do lots of things with linear models. Like this has been shown very extensively in the new brain imaging literature. With careful feature extraction followed by linear models, we can do a lot of things with like state-of-the-art and very interpretable models. So that should be acknowledged. So I mean, interpretability can be at reach in certain cases if the system was designed in a way which makes it so.
If I just may say a word. Oh, sorry. Yeah, I completely agree. And to me, it's also about acceptability for the patients and the doctors. Because even if we have the best algorithm ever, if it's a complete black box, even the patient or the doctors might be reluctant to use it. So even above that, just for AI to really make its place in the medical world, I think explainability is very important.
I completely agree.
So thank you very much for this very interesting and valuable discussion. So we are now going the step ahead with the program and switching to the second session. The second session will start with Dr. Pritesh Sethi from computational biology from IIIT in Delhi. So in the Prasta Institute of Information Technology. And so I'm really glad to have you there, and please.
Thank you, Jan. Thank you, Professor Mikkel Paul, for giving me this opportunity. I'll just start sharing my screen, and please let me know if my slides have come up.
Yeah, we can see it, no problem.
Great. Okay, so I'm in the presentation mode. Please let me know. I'm going to switch slides. Please let me know if I think it's still coming up. So do you see my slides moving?
Yes, that's moved. Yeah, perfect.
Perfect. Thank you so much. So I'll be talking about real-world AI solutions for healthcare. I come from background in medicine. I did my medical graduation, and then I decided to pursue research. And for the last 15 years or so, I've been working in the space of data and models, and working very closely with the healthcare settings. I've been associated with healthcare institutions, including the All India Institute of Medical Sciences, New Delhi, where I'm a fellow of the Wellcome Trust DBT India Alliance. And there we run a project in the ICU settings. More recently, we have started an initiative at IIIT Delhi, which is the Center of Excellence in Healthcare, which aims to basically address these gaps that are missing from data and modeling side for healthcare. The infrastructure gap, where we don't have adequate platforms and digitalization, recording, reporting, planning, and including new devices. We have a knowledge gap where our skill for the workforce is not skilled. The students are, we don't have that bridge expertise between medicine and computer science. So we design courses, and I teach a few courses in that direction. And then, of course, we engage with the policymakers, including the governments, in addition to our research, where we build some of these platforms and models that can help take decisions. So I'll jump into my lab's research, specifically in the directions that I've planned for the next 10 minutes or so. So the first is computer vision and signals. And here, this is a project which we are implementing in the Department of Pediatrics at All India Institute of Medical Sciences, where we realize that the ICUs are data-rich environments. These are the largest and the densest source of data routinely generated and routinely lost. Depending upon the beds, number of beds in the ICU, it could range from anywhere between 1 GB to 3 GB per day, which is lost routinely because we don't warehouse and then we don't build models on top of these. Typically, these data get overwritten every 24 to 48 hours. So this is a project we call Sepsis Advanced Forecasting Engine, the SAFE ICU, supported by the Wellcome Trust DBT India Alliance. And in this project, we have warehoused more than 1.5 million patient hours. This number is a slightly older number. More than 1.5 million patient hours of continuously monitored vitals data, which we have now paired up with treatment charts and laboratory investigations data to start building models on these data sets. So when we started really lean. We started by actually building communication protocols with using the communication protocols with the monitoring equipment and so on. And that allowed us to build in quality initiatives that every time a bed stops sending us data, we receive a mobile phone alert because we were building this from the ground up. And then we started very lean with value that we built for clinicians. We didn't right away jump into AI models because we wanted to build trust first. And in building dashboards and questions that clinicians were asking, for example, how is the antibiotic usage? in my ICU? What is the data quality? Which bed is giving good quality versus poor quality data? Is my baby in the right oxygen saturation range or not on a given bed? Because babies can go blind if the oxygen is more than 95% saturation, or they can go into hypoxia if it is below 88. So developing these data science sort of metrics for doctors gave us more confidence and gave them more confidence in helping us build predictive models going forward. And then we really started building the AI models, where we developed cohorts such as shock prediction, sepsis prediction, organ failure prediction, and so on, where we basically predict what is going to happen in the future. So when we say prediction, we are not looking at phenotyping or detection, we are looking at prediction in time. So one of these examples, models where we used computer vision and signals was prediction of shock up to 12 hours ahead of time. And at that point of time, we did not use deep learning. We used SIFT-based and standard computer vision approaches to figure out features. And these features were clinically driven. For example, every first-year medical school student knows that if hands and feet are colder and abdomen is warmer, it's a sign of redistribution of blood. So we use this medical domain knowledge and understanding to build features and feed them into longitudinal models, where we were able to predict up to 12 hours ahead of time. Now we have actually built deep learning models which can use videos to predict outcomes such as shock in the ICU. The paper is under review right now. And also another paper that's under review talks about generalization of these models, because we believe that models that are built in a setting need to also generalize. So there we extensively use deep learning approaches and feature selection approaches in tandem to build models that were generalizable across 200 different ICUs across the United States and Indian settings. Another computer vision model that we recently it got accepted for publication in JMIR Public Health and Surveillance was a model that looked at mask usage on social media and its patterns with policy changes and movements such as Black Lives movement in the United States. So we looked at data from the United States and we built models which looked at mask use and mask fit. So we developed some facial indicators to basically guide our models and use social media images to understand what policy interventions, what was the impact of policy interventions in different states in the United States, and what were the patterns which were associated with those. We believe that, I mean, we always complain in healthcare that we have less data, but I believe there's a lot of data out there which can be triangulated, combined together and put together and to build meaningful insights. And in that direction, I will just show the next set of slides, which will talk about language models. So our lab in the last 1 year or so has been very active in language models for healthcare. And this started with one of the models that we call Wash Karo. This started before COVID, where we wanted to build models that can look at WHO guidelines and look at what's happening in news, use language models to summarize and to select the right information and to deliver it to common people who basically then can consume that information, because nobody reads WHO reports. So summarized tidbits of information in local languages such as Hindi at the right time. So right information to the right people in the right format at the right time was the goal of Wash Karo application. Karo means do, so do wash. This became really important at the time of COVID-19 pandemic, where we repurposed this application for COVID. And this engine summarizes how it was done. Essentially, we looked at authenticated sources of information such as the WHO guidelines and also the Ministry of Health and Family Welfare guidelines. We looked at what was coming up in the news. We summarized these using state-of-the-art AI summarization models. And then we generated similarity scores, matched them. A team of clinicians actually vetted about 13 different scoring metrics to choose which is the best scoring metric for similarity. We use that metric to select articles and then use translation and text-to-speech APIs to deliver this information to the public. And this was available as an Android application. A feature there being that users could give feedback on thumbs up or thumbs down on whether the information was useful to them or not. This is recently accepted for publication in scientific reports, but we were able to show in a short period of time that the thumbs up and thumbs down actually led to our AI engine learning and presenting more relevant information over time, as is seen in the left graph. It also helped us to segment health information, for example, more females engaging with the app in Hindi than males over the period of time that we were online with COVID. Unfortunately, Google's policies changed, so only governments were then permitted to spread COVID information, but we repurposed our app for tuberculosis, and now we are actively rolling this out, a very similar public health intervention out for tuberculosis. We also have worked with language models that have mined recent literature because COVID literature have been rapidly emerging. So we said that if, for example, hydroxychloroquine literature came up and went away, it is not emerging evidence. So what comes up, stays, gathers more evidence, and becomes heavier in terms of these streamlines that you can see here in topics, was the core idea behind our pipeline, a data science pipeline, which we call evidence flow. And the way this pipeline works was essentially we looked at WHO vetted literature, and this is ongoing. So we have now recent updates on the literature as well. We used deep learning models and embedding models to learn embeddings on these literature. And then basically we built networks on the top of these embeddings. So because these embeddings will keep on changing, these are dynamic. And then we looked at these networks to see how communities emerge. So, and can we predict what theme is likely to emerge in the next few months or so. So this paper is currently under review, but the dashboard is online. It is called Evidence Flow, and the preprint is online. You can go and read. We were able to show ahead of time emerging themes such as coagulopathies and neurological complications and evidence for them getting stronger. So this paper is under revision right now, and we hope we'll be able to share more details on these. We've also used reinforcement learning in our recent research, where we asked this question: can optimization using reinforcement learning be used for resource allocation in healthcare settings? And one of the models that we built was for COVID-19 vaccine allocation. We started out in August of 2020 when there was no vaccine available, but reports had started coming out that vaccines are going in first phase or second phase. So essentially, we developed a contextual bandit model combined with the actor critic model, a deep reinforcement learning model, which actually we were able to train to basically then show that if we used an intelligent strategy, we will always be preventing a positive number of cases than beyond a naive strategy or the current strategies. And that depends upon the stage of the pandemic that we are in. But we believe that this is a generalizable approach. And now we are building a plug and play setting for bringing more democratization of reinforcement learning in public health resource allocation. This is in advanced stages of revision. We hope it will be accepted soon. But the dashboard vaccine is available and preprint is also available. Yeah, so these are the different kinds of algorithms that are available. Finally, I'll just finish by saying that a question came up about explainability in the previous session. I am a strong believer, coming from a medical background, that if our models can explain, they will get much more adoption and trust. So we have been building explainable AI models of very simple kinds like Bayesian network models, because they allow us to engage with the model and then basically query these models. But structured learning, which can be achieved using Bayesian networks, could be built using deep learning based methods, is one of the themes in our labs. And we have been showing the application of structured learning models in various public health scenarios, such as what should we do to minimize respiratory illnesses in India. This was published way back in 2015. These are a few other papers that we published using similar approaches. Health inequality, social determinants of health in the United States. This was in AAAI. More recently, learning the mental health impact of COVID-19 in the United States, and also looking at interventions that can be rolled out for HIV in the 5 different states of India. So I'm aware I'm running out of time, so I will stop here. That's my team and my support. I would like to thank all my team members and my colleagues who have been in this initiative. Thank you so much, and I'll be happy to take questions later.
Thank you very much. Yes, exactly. The Q&A session will be just after the second talk of this subsection. So this second talk, thank you very much again, Dr. Tavri for this valuable talk. And so now we are switching to Gaël Varocco, who is a research director at INRIA, which is the National Institute for Research in Computer Science in France, and so is belongs to the Neurospin Research Center. So thank you again, Gaël, to accept this invitation, and the floor is yours.
Hi, everyone. Thanks a lot for being here and for the invitation. So my talk isn't actually going to be on scikit-learn. I sent my information a bit late. It's going to be on electronic health records and specifically actually on dirty data. And I'll show you the link between those two things. The first thing I'd like to start with is the role of electronic health records on medical research and medical practice, and the growing role. And to start with medical research, so the gold standard of medical research are clinical studies, but clinical studies are extremely hard. And if we think, for instance, of the COVID-19 vaccines, it took much longer to actually develop those vaccines than to test them on real human volunteers. So the reason why it took much less time to develop them than to test them. The reason it took less time is, of course, because there was a lot of work before, and basically the technology was available to get started. However, we need to keep in mind that actually evaluating the drugs and the treatments is a bottleneck in medical research. The reason is because experimentation on human is difficult, it has slow cycle, high risks, and because the conclusions that we need to draw need to be drawn across individual heterogeneity, and that is hard. So we can't get real life evidence, and this real life evidence is complementary to clinical trials. Some questions, for instance, such as do vaccines prevent spread, are difficult to study with clinical trials because the intervention is challenging. It's not impossible, it's challenging, and it requires a huge sample size. Clinical trials may have problems that are known of external validity. By this, I mean that they are done in a certain setting on a certain population, and then we need to generalize them to different settings and different populations. One example is the AstraZeneca vaccine, which in the original trial was only experimented on non-elderly people. And so we can't conclude on whether it's applicable to elderly people. So For these questions, it may actually be more beneficial to use what people call real-world observational data, which is to follow individuals as they get or not the treatment rather than an actual clinical trial. So electronic health records give us a very interesting source of real-life data. What I call electronic health records are basically patient records, anything that's available. Reimbursement claims databases, accounting, the history of measurements, the doctor's notes. They're amazing because they give us a great longitudinal coverage. So for instance, in the network of hospitals in Paris, we see 8 million patients a year. So it's an incredible epidemiological sample. And another benefit is that it's almost free data because it's data that is acquired anyhow during medical practice. Now, the challenge is that this is very complex data. It's data that was not acquired for a specific analytic purpose. It's spread across multiple tables with missing information. It's inhomogeneous. So we have uneven data across patients, across hospital sites. You know, some patients have different measures because sometimes it's not applicable, because sometimes it's more challenging to make the measures. So compared to a clinical setting, it's a huge rate of missingness. I would like to say that the notion of missingness is not the right one, because we're lucky when we have the data, some data available for a given individual. And then the challenge is that the information is not normalized in any way. Often there is manual input with different conventions. We have, we might have strings that are used to relate to the same pathology, but varying strings. And the problem is actually even more complex because the data may be represented, spread out in different ways in different tables across the hospitals. Now, another challenge of electronic health records is that we're looking at observational data, not experimental data. Doctors are prescribing treatments to patients, but this prescription actually carries a lot of analysis from the doctor. So treated and untreated patients are often non-comparable. So we can't draw naive conclusions on treatment efficiency. And that's a classic problem of causal inference. And there are a set of techniques that can account for the baseline status of the patients to compensate for those differences. And for this, we can use machine learning models. We can, for instance, learn to predict the outcome given the treatment and do this with machine learning. And then we can use this prediction to compute the efficacy of the treatment. We can learn to reweight the patients to give more weights to unusual patients that were treated or unusual patients that were not treated. And this, again, this can be done with machine learning. So this was a short introduction to the stakes of electronic health records and the fact that the challenge in the electronic health records is that they're not curated data as we would like to see them. And now I'd like to show how machine learning, advanced machine learning, can come in and fill this gap and analyze these challenging data. Now I'm switching to a slightly more formal part of my talk that is not directly applied today to health. And I'll first start by talking about non-normalized data. So to take an example in employment, you might have a column of employee position title that is non-normalized with lots of variations. Now, standard statistical practice treats the different entries or separate as non-related entries, using, for instance, Wahao encoding, and it breaks with such data for many reasons. So the standard approach is then to try to normalize the database. And for this, one approach is to do some form of feature engineering to try to get a more regular data representation. Another point of view is that of deduplication or record linkage, which is to say we're going to try to merge entities that are related. Now, these approaches require a lot of manual intervention. They're very hard to make automatically at turnkey. And our point of view on this problem is that we should be doing supervised learning directly on the dirty categories. And one of the reasons is that the statistical question at hand should inform curation. It's not obvious to know when you should be merging two different strings or when you should not, and that might depend on the underlying statistical question at hand. And so the first thing we did here a few years ago to explore this idea is to replace one-hot encoding with a soft version of one-hot encoding, which accounts for the string distances. And now that's a trivial idea, but what we've shown empirically on many real-life databases is that accounting for the strings actually improves the analysis quite strongly. It gives much better statistical analysis. Now, the problem here is that these approaches basically anchor the approach on a small number of strings, prototypes. And that doesn't scale well. So another point of view is to say what's interesting is a set of substrings. And those substrings are basically giving us the underlying reference categories. And so then what we did is that we did topic modeling on substrings. So basically modeling strings as linear combinations of substrings. And so that's basically a matrix factorization. And if we do the right model and we work on the right model, what we can achieve is something that takes readily the raw strings and basically extract latent categories. But those latent categories are coded in a continuous way. So each string is coded as a combination of a small number of latent categories. And so this gives us automatic feature engineering. And the nice thing is that it can be made understandable to the user because we can extract the recurrent substrings. And then we can use black box AI interpretation tools. And so we can have a full pipeline that's automatic and that's understandable in the sense that it relates to concepts that are understandable by the user. So this is available in software, Dirty Cat. You're welcome to try it. Now, another challenge that we have is missing information. This is a challenge that is well studied in classic statistics, where the model is that we have a complete data generating process and we have a random process including the entries. And under certain assumptions, under certain statistical assumptions, you can have basically very powerful theoretical results that tell you that you can ignore the missing values and use techniques such as imputation or EN and still get a valid answer. Now, those techniques break down when the missingness is not ignorable, when there is some form of, for instance, censoring process that you can see here. Those techniques need ignorable missingness. Now, we're actually interested in a different problem. We're actually interested in predicting with missing values, because predicting with missing values can be used, for instance, for causal inference, for prognostic models, for diagnostic models. And now the problem is slightly different. We're not trying to recover a model that is valid, the model of the complete data generating process. We're trying to do the best prediction, accounting for missing values. The difficulties here is that we have basically a combinatorial problem because we have a half discrete impute space. And what we can show theoretically is that even in extremely simple settings, the best predictor is extremely complicated. And the intuition here is that each time a data point is missing, we need to compensate for this missingness. So we have 2 to the power D set of combinations here. And so what we did recently is that we derived an optimal neural network that approximates those 2 to the D potential predictors. And for this, what we did is that we derived the theoretical forms of the optimal predictors, and then we looked at functional approximation theory to build a specific a network that is optimal for this setting. And so what we can see, so we call this network the Numis network, and what we can see is that it needs much less parameters to approximate well the optimal prediction. And so this means that those networks need much less sample than things like MLPs, where we replace missing values by plug-in values. An interesting reason here is that unlike imputation, those networks work well even in missing, not at random settings. So we can use them on real life data where the missing value is somewhat adversarial, which is absolutely the case of electronic health records. So to sum up, electronic health records, I strongly believe they can open new doors to cheaper study, to better prognostic, to better diagnostic. But they're extremely challenging to use, and modern AI provides the statistical estimators and the information extraction that is needed for this. And I've shown you two applications. One is the ability to learn from categories that are represented with uncontrolled strings. And for this, we have readily available software. And the other one is how to do supervised learning with missing values. And I'd like to stress here that the trade-offs are completely different than the trade-offs in the classic missing value literature. Thank you.
Thank you very much, Gael. So just in time, perfect. All the speakers are really respecting the timing. So thank you for that. Thank you for all this interesting project that you present, Gael. And so now, if Professor Sethi can bring on so his camera, maybe we can go to the Q&A session for the last two talks. So if someone wants to raise a hand or unmute their microphone to ask some question of the two last speakers. So Jan, do not hesitate, please.
Okay, hello. I'm just trying to put the video as well, but it doesn't seem to work in the browser. Anyway, thank you very much for the two talks. I have two questions for Gael. First, how do you choose the optimal size of the substring, the length of the substring that you're looking for?
Well, thanks for this question. So the way we work is that we work with character level n-gram, and also we have a set of stop characters which help separating words. And so we have a mixture of these. And so empirically, I didn't go into details, but part of this research was really driven by a lot of empirical validation. I think we used a bit short of 20 real life data sets to validate things. And empirically, we find that typically it's useful to take character level anagrams from 2 to 4 characters, and to also add words that are separated by commas, spaces, dots, and everything.
Okay, thank you very much. And the second question that is much more open, I think. So you were discussing about missing values. How would you handle as well value that is not missing, but that is of low quality? For instance, you have some required fields, and the doctors might put a nice score, but sometimes they might put just a 0 just to go on with the process.
If you say just a 0, I think you're basically alluding to something that would be some form of like a systematic outlier. So one thing that we're really discovering as we move forward with this research is that we need very flexible statistical models. Typically, if I want to advise people on using something that's already available, I will advise them to use things like gradient boosted regression trees that are extremely powerful. And so we need to use statistical models that can deal with irregularities, with a predictive function that has discontinuities, things like this. And if you have enough data, now that's some form of a theoretical answer, but if you have enough data on such kind of a problem, the model will asymptotically converge to the best possible prediction, which is the conditional expectancy with the data. And I like to say this seems to be like an easier problem than the missing value, even though of course the missing value can be brought back to this problem, because here you're still hopefully you're still in a continuous space. The real problem is discontinuity. Now, if you have a discontinuous behavior from the user, then of course you have a challenge.
All right, thank you very much. Yeah, the problem is that the 0 could be sometimes a score that is indeed 0, and sometimes just a filler gap, and you don't know which is which. See what I mean?
I think the right answer then is in the UX And I think, from the big picture, we should be changing our UX so that people don't do that. Because if if you have this collision of information, you're just losing information. You can't do anything.
Thank you. Do you have other questions? So from my side, I will have the same question than previously for the previous session. So I will start with Professor Sethi. So Professor Sethi, if you want to imagine the future in the next 5 years, yes, it's quite impossible, but imagine that you will have an infinite funding. What should be the next step that is, in your point of view, the most important and unavoidable for the research in your field?
Yeah, thank you. First of all, thank you, Gail, for that wonderful talk and bringing up the challenges that are there in the medical data set. So I think, yeah, going forward, I believe the key problem in healthcare is reliable data sets, and how can we actually build more research into directions which allow learning across data sets. So federated learning, building models that can generalize across different settings. How do we exchange models in a way where federation can happen without compromising patient security and patient privacy preserving technologies and all of that. So I believe that that is the key thing, because unlike other areas, healthcare data are pretty siloed as of now. So how do we break these silos and build architectures, computational models that can actually enable us to ask bigger questions? I believe that is the key area I would be very much interested in going forward.
Thank you. Thank you, Professor Sethi. Same question for Professor Varocco.
I think basically we need forms of successes. I'm a doer, I guess. I want to jump on problems and solve problems, and by solve problems I mean convince practitioners that we're bringing value to to healthcare. Because one of the big problems we have is a problem of trust, and this problem of trust is a trust that is coming from all sides. Practitioners don't trust us, patients don't trust us. And, sure, it's legitimate. It's not even trust, it's value. They don't see the value. So Professor Sethi was mentioning the problem of siloed data. Well, I'm lucky not to have this problem with very big hospitals that we're in, but I think this problem of siloed data will get much better if all the different stakeholders believe in the process. Right now, they don't believe in the process, and so they're not helping us. And if they believe in the process, they will help us. It's been my experience in the past. And for them to believe in the process, we need to give value. So, a big problem, I think a big problem is getting stakeholders together and solving all the little problems to have successes. I don't want to comment on something bigger than this. That's already pretty big.
Thank you. Thank you very much. Nikhil, do you have a last question for our speaker? Nikhil, do you hear me?
Yeah. No, I don't have any question for them.
Perfect. Thank you. So thanks again for these two valuable talk. Thank you very much to be there this morning or afternoon in India, and to participate to this session for the Knowledge to Me 3. So we are now going to the third subsession of this morning, afternoon, Thursday. So thank you very much again to both of you, and we are now we'll have 3 different talks for this last third subsession before the last Q&A and general discussion session. So let me please welcome Dr. Pratosh, who will be the next speaker.
Hi.
Hi.
So
Hi. I was not sure to see you. Okay, perfect. Thank you. Thank you to be there. So Professor Pratosh is assistant professor in the Department of Electrical Communication at the IISc Bangalore, so Indian Institute of Science in Bangalore. And so I'm very glad to have him today. And so if you can share your screen and your slides, you can start with pleasure. Thank you very much. Floor is yours.
Thank you, Professor. Sorry, I heard somebody speaking.
No, go on, go ahead.
Okay. Yeah, so I assume that I have 10 minutes or?
Yeah, maximum 13 minutes, please.
Okay, sure. Let me just set my stop clock so that I'll not spill over. So timer. Sure. Okay.
So for information, we are not seeing you neither your slides.
Sure. Let me just share it. Yeah, give me a second. View, no. Share screen. Yeah, do you see it now?
Yes.
So maybe I should make it full screen, right? So
yeah, let's try it, please.
Is this better?
Yeah, please try to move it. Move one slide to another. Just okay, it works. Perfect. Thank you.
This is good. Okay, thanks. Okay, so it went to some other view. Okay, yeah. So good morning to my French colleagues and good evening to my Indian counterparts. So I'm Pratosh. I'm a faculty at Department of Electrical Communication Engineering at IISc Bangalore. So I just moved to IISc Bangalore a month back. Before that, I was at IIT Delhi. So Dr. Sethi knows me. We have met multiple times. So yeah, so I've been looking at representation learning for affordable digital health. This is I'll spend the next 10 minutes in sharing some of the thoughts that I have and some of the challenges that we faced when we try to solve these kinds of problems. I also happen to be the co-founder of a small company with my student called Cognable Tech, where we try to use computer vision and deep learning to aid children who have been diagnosed with autism, basically on behavioral healthcare. Yeah, so the work that we have been doing, can be classified into three buckets. One is the diagnostic healthcare, where we develop methods to aid clinicians in diagnosis. The basic idea is to use machine learning methods to predict the clinical parameters from various type of data sources. And you have this predictive healthcare where you build techniques for prediction of unseen clinical events, right? I mean, I've been working on a lot of time series kind of data where you have data coming from the bedside monitor and you predict the events that might happen later in time. So the third bucket is the palliative or assistive healthcare, where you develop tools for managing diseases and therapeutic interventions. And the goal of our startup also happens to be falling in this category. Yeah, so I'll give a few examples of what we have been doing in all three buckets. So in diagnostic healthcare, before coming to academia, I was working with the Xerox PARC, and where there we developed a bunch of algorithms where we try to estimate the human body vitals, such as the heart rate, respiration rate, and so on, using a completely non-contact and non-invasive method by using a consumer-grade camera. So the idea is that you just have a camera in front of the subject, and you take a video feed and you try to extract the heart rate and respiration rate from the subject. So we did a bunch of experiments with Stanford NICU, and one good use case of such methods is in the neonatal ICU, where it's it's very difficult to have a lot of probes on newborns and infants. So I mean, the idea was to use a consumer grade camera and still get a lot of human body vitals estimated. So that's one area that I looked at. The other area that I'm currently looking at is a differential diagnostics in pathological and histopathological images, that you are given pictures from microscope taken of different body tissues, and you solve the classical computer vision and machine learning problems, starting from the T-blurring and classification and regression and whatnot. The other problem that I'm looking at is classification, segmentation, and localization problems in histopathological and radiological images. And in a country like India, there's no dearth of getting data. You get huge volumes of data, and there is a large need to initially screen these kinds of images for the presence or absence of certain kinds of ailments. So we look at large chunk of computer vision problems on histopathology and radiological images. So this is what we broadly do in the diagnostic healthcare space. In the predictive healthcare, the aim is that in diagnostic healthcare, the idea is that you have whatever the clinical event or rather the marker that you are trying to estimate is already present in the data that you have been given. But in predictive healthcare, the clinical marker that you would want to present is not apparent in the data that you are given. For example, I've been working with a hospital where we wanted to estimate or predict the stability of spine using X-rays. Now, the clinical gold standard is to use an MRI scan to estimate the stability. But this doctor told me that in a country like India, not everybody can afford MRI. There are some hypotheses that would say that perhaps we can estimate the stability of a spine post injury just using X-ray, which is affordable. The other area that we are looking at is trying to look at octa images. When a person comes for an eye scan, there is some evidence to show that the cardiovascular patterns in those blood vessels can tell you, the blood flow patterns in those blood vessels can tell you a lot about the cardiovascular risk that a patient has. This is something that is not apparent in the data that you have. We're also working on risk analysis or early warning systems from clinical time series. One example is that if you are given the data that is recorded from the bedside monitors within an ICU, we would want to predict episodes like acute hypertension episodes and the episodes of stroke and heart attack and so on much prior to their possible occurrences. In the space of palliative healthcare, I already mentioned about the startup that we are running. So what we do is, we record a bunch of videos of a therapeutic caregiver who is interacting with a child that is diagnosed with autism, and we measure the response to certain stimulus, and depending upon that, we offer a customized or an individualized treatment plan to a given child, and we use a bunch of computer vision and deep learning algorithms such as action recognition and behavioral analytics doing this. We also are looking at video cameras as continuous biosensors. So the idea is that suppose you are having a Zoom call like this. This is the holy grail of telemedicine, where you are talking with a doctor or a clinician via a Skype or a Zoom call. While you are doing it, all your body vitals are being estimated in a non-contact fashion, and the clinician has access to that. We are also looking at a lot of speech and language therapy, where we try to get some laryngography type of parameter estimated from speech signals. So these are the gamut of problems that we are looking at. But what's interesting is that when we take up each of these problems individually, there are all kinds of machine learning problems that one can think of that would emerge in these kinds of problems. See, this is very unlike other non-critical applications where you would want to build a recommendation system for an online store or something. This is critical application, and you need to be doubly sure when you are building these machine learning algorithms. And these are some of the problems that I have encountered on a personal in a personal level. See, one is unavailability of large scale annotated data. We all face this problem that thankfully in India, there is no dearth of data per se. When I talk to doctors, they'll say that, you take as much data as you need, because we are always overburdened by the patient load that we have, but they don't have annotated data. So you need expert annotators, and you have missing and noisy data, right? I mean, you have things like sensor misplacement or wrong annotations that are happening, and lots of label noise that is coming in the picture. One other problem that boxes all the time is the domain shift or inductive biases problem, right? I mean, I work with certain startups. One startup that I work with, what they did was they built a classification algorithm for classifying the images of WBC, right, into some of the categories. When they shipped their algorithm, I mean, in one of their customer centers, it stopped working. The algorithm that was giving about 90% accuracy suddenly started giving around 30% accuracy. When they probed into it and saw what the issue was, the only difference was, the other center is just using a different type of microscope. So just by changing the camera lens, your algorithm stopped working, which is the very well-known problem of domain shift. And you have the problem of heterogeneity of samples and class imbalance, right? Because by the very nature of healthcare, the data sets that you get are highly imbalanced, because if you take 100 X-rays, the ratio of malignancy to the benignities is always skewed. So you can't just simply take a off-the-shelf ML algorithm and apply there. And you also have the widely discussed non-interpretability of the deep learning models, which is the black box nature of the deep learning. And some of the things in some of the applications, what I've seen is, you need very careful calibrations that are to be done. One example is when we were trying to estimate the peripheral oxygen saturation, or SpO2, from videos. We could very well do it when we could very well estimate the heart rate and respiration rate using videos, because they both are frequency measures. You don't have to care about the absolute amplitude. But the oxygen saturation, if you want to get it from a simple video, you need to calibrate that with the pulse oximeter that is given, and calibration is a big problem which you cannot do without multiple sites. So I'll just give a small example, before I conclude. So this was one problem that I just mentioned about. These are the images of WBC that they were categorizing into 10 or 11 categories. So these are, every row what you see is the images from a particular camera. So as human beings, we actually did a Turing test sort of things, where we gave these images from different cameras to human pathologists, and they had no problems whatsoever in classifying them properly. But if you have a machine learning algorithm that you have built on images from camera A and test it on the images from camera B, it fails drastically. So here is a classic computer vision example, where the first row that you see is images of horses. We recognize all of them as horses, irrespective of the kind of data set that it comes from. But you train an ML algorithm on one of the data sets and test it on the other, it fails. This is the well-known problem of domain shift. So we recently came up with, because I thought the talk should be a little technical as well. So we came up with an algorithm recently to solve this problem of domain shift for the clinical images. So the idea was simple. What we said was, as human beings, we always have this tendency of mapping something that is unknown to something that is known, right? I mean, if you are shown something that is not known, the first instinct is to relate it to something that we already know. Now, we said, okay, when you are given an image or data from something that is unknown, which is the target data set, you just simply don't classify it. But what you do is you try to map it back into something that you already know, which are the samples of the source data set. So, skipping the details and the math, basically what we do is, given a target sample from a data set that is unknown, we would try to search in the space of the source data by solving an optimization problem on the generative model that is built on the source data, such that you find a clone, so to speak, in the source data set that is very close to the target sample that you are given. Right. So here is an example. The figure to the left of the screen is what you see on a general type of image. The first image is that of a chair, right? That is from the target data set, which was misclassified. So now what we do is we solve that optimization problem, and from a chair that is unknown to the classifier, we stop at another chair, which is semantically a chair, but it is completely different than what the initial setting was. So this chair that you get after you do whatever the solve the optimization problem is close to the source data set, and therefore easily classifiable using the classifier that you have trained on the source data. So this is the application on the problem that we actually encountered. So you have a target data that is given from one of the cameras, which we call the camera B, and when we solve our problem, the semantics are the same, but as you can see, I mean, the stain and the background has changed to be closer to the source data set, so that the accuracy of the classifier increases. So yeah, so what I think one can do, I mean, we can do as a community of people who are trying to bring in ML to healthcare, is that learn meaningful representations instead of just throwing a pre-trained model onto the problem, the healthcare problem. See, what I mean to say is that we all know that imposing certain type of regularizers or priors while you learn drastically changes the representation that is learned. So what I have realized is that we should try to emulate the clinical practitioners, in the sense that when I talk to these doctors who give me these problems, the first question that I would ask them is, how would you solve this problem, right, as a clinician? And they would tell that, look at this image, there is some sort of a shape here, or some sort of the gradient that is seen here, and all that. So the idea is, while you use the state-of-the-art and off-the-shelf machine learning algorithms, you try to impose the clinical knowledge as a sort of priors in your algorithm, so that it both becomes explainable, and also some of these problems of domain shift, etc., will be taken care of. Yeah, so these are representations should be learned with clinical knowledge-driven priors and regularizers. The other thing that I'm looking at, that our group is looking at, is build generative models to generate synthetic data that are explainable, identifiable by constructions. We all face the problem of not having enough images, so synthetic data can be one way to do it. See, one other thing that I always talk about whenever I present these things in fora like this, is that maybe somebody should try a BERT or an ImageNet kind of models for clinical data sets. People are coming up with such kind of data sets for speech, images, and all other types of modalities, but we still do not have an ImageNet level data set for, say, pathology or histopathology. I think a multi-center, multi-country, or even multi-continent kind of consortium would be needed to create such a data set, and also the models that will be made publicly available. That would be sort of learning representations that are spanning across different sites and different organs and so on. So I think with that, I would stop here, and I thank the organizers for giving me an opportunity. It's a pleasure to be presenting here. Thank you.
Thank you. Thank you very much, Pratesh. Very interesting talk. So we are actually just on time. So I will give now the microphone to Cedric Gongloff, which is from the University of Rennes, from the Public Hospital Research Center. And you will have, as the other, please try to remain in the 13 minutes of the presentation. Thank you very much.
Okay, thank you, Jan. You can hear me, I think?
Yes, perfect.
Maybe?
And we see your
okay.
slides.
Okay, and are they moving now?
Yeah.
Okay, perfect. Thank you very much, Jan, for this introduction. As you said before, I'm a member of the LTSC laboratory in Rennes, France. But my main job, okay, I'll start my video. Now you can see me. My main job is to work in the emergency department in the same city. So I'm not a data scientist, I'm a doctor. And I will present you a study performed during the first episode of the COVID-19 pandemic, showing that machine learning can optimize COVID-19 diagnosis by using common clinical and biological variables. Concerning the background, increased mortality and sometimes the collapse of entire healthcare systems have been reported in several countries in the beginning of the pandemic. And indeed, due to COVID contagiousness, promiscuity can promote patient to patient and patient to healthcare workers transmission, leading to the saturation of health systems. To limit this effect, COVID-19 patients are hospitalized in specific units after being triaged in emergency departments. Therefore, it is essential to have an effective and easy-to-use tool to diagnose COVID-19. PCR is a standard test for COVID-19 diagnosis. However, like for any other test, there are false negative results. Therefore, additional investigation should be performed in patients with negative PCR, but with high clinical probabilities of COVID-19. In this case, chest CT is a very interesting tool because it allows detecting virus-induced lung tissue damages. The combined use of chest CT with PCR allows to resolve diagnostic ambiguities and to avoid false negative results. However, it is very expensive and time-consuming to perform both PCR and chest CT on every patient to avoid false negative results. So the main objective of the study was to build and evaluate machine learning models to improve COVID-19 diagnosis. So the predictive variable was the presence of COVID-19. COVID was diagnosed as follows. Hospitalized patients were examined by a physician who estimated the clinical probability of COVID-19. PCR and chest CT were both performed in all of these patients, and COVID was diagnosed by two ways. First, when patients had positive PCR, they were considered COVID positive. And the second way, when clinical probability was high and chest CT showed typical COVID images. The patient was then considered COVID-19 positive, even if PCR was negative. In this case, PCR was considered a false negative result. Patients were then allocated to the COVID and not COVID group. Concerning the predicting variables, all variables present in our database were collected. This represented about 3,000 of variables. They were essentially divided in two fields, the clinical field, such as everyday treatments, symptoms, etc., and the biological field, such as blood tests, number of white cells, inflammation markers, lactate, blood levels, etc. And the data were collected from a local database called EHOP, in which health data are integrated and identified in real time. Structured data, such as biological results, were directly collected, but we had to structure text fields by using regular expression before model building. In order to simplify our data sets, we decided to reduce the number of variables used for model building. Concerning numeric variable, we used student t-test to compare means between COVID and not COVID groups. And for binary variables, we used the chi-square test to compare proportions. And variables with a p-value below 0.2 were considered as variable of interest. Comparisons between COVID and not COVID groups showed that the variables selected for model building were similar to COVID-19 signs. These variables belong to 5 categories: clinical signs, blood gas, blood cell counts, ionogram, hemostasis, and liver enzymes. We only showed clinical signs in this presentation. And as you can see, the proportion of these symptoms was higher in the COVID than in the not COVID group. So variables selected for model building were very consistent with commonly reported signs of COVID-19. To avoid multicollinearity, correlation coefficients were calculated for each pair of variables. And when the coefficient was higher than 0.8, one of the two variables was excluded from model building. Therefore, the final set of variables selected for model building included only 22 variables among the 3,000 initially present in a complete database. And the machine learning models were built with RStudio and the Carrot package. We created 3 types of models: neural networks, random forest, and logistic regressions. And each type of model was built in 2 versions with 2 sets of variables. The first version was only built with the previously reported 22 variables. For memory, this corresponded to basic clinical signs and routine biological tests. And the second version of each model was built with the same 22 variables, but included in addition the result of the PCR. So concerning the machine learning models building, we started with the whole data set, and data were randomly divided in 2 parts, the training and the test data sets. The training set corresponded to 80% of the patients and was used to build the models. And a k-fold cross-validation was performed in order to prevent overfitting. Overfitting occurs when the model captures the noise of the data, and in this case, high performances are observed on the training data, but poor results are observed on new data. Concerning the k-fold cross-validation, the dataset is divided into two k parts called folds, and the model is trained by using k-1 folds, and the remaining folds in red is used to validate the model. The same procedure was applied k times, and in this study, 10 folds were used for model bridging. Model performances were assessed on the test data frame, and to evaluate performances, models prediction were compared to the presence of COVID-19 in the test data frame. ROC curves were constructed accordingly, and the area under the curve was used to evaluate models performances. As a reminder, area under the curve will vary from 0 to 1, and a perfect model has an area under the curve at 1. So you will find in this figure the performance of two machine learning models that we constructed, and they were based on logistic regressions. The first one appears in black, and was built only with the 22 common clinical biological variables. And the area under the curve was at 0.77. As a reminder, this model did not use PCR nor CT scan to diagnose COVID-19. So it is possible to diagnose this disease with such models when you have no PCR or no CT scan. But performances are clearly not optimal. The second model, represented with a red curve, was based on the same 22 variables with the addition of PCR and had an area under the curve at 0.93. So it was a pretty good and almost perfect classifier. And this is very encouraging because we also evaluated the performances of the reference test, which is RT-PCR alone. And we found that the area under the curve for PCR without machine learning was about 0.85, which is not perfect due to the presence of false negative results detected with chest CT in this study. In conclusion, this study demonstrates that machine learning models can be developed in order to improve COVID-19 diagnosis. And such models can increase the performance of PCR by using common clinical biological variables. We built other models in this study that also improve chest CT performance to diagnose COVID-19 without PCR. So if you're interested in this topic, I encourage you to read the paper that was published last year in the scientific reports. And thank you very much for your time and your attention.
Thank you very much for this very interesting talks and very interesting results. So thank you. I recall for the audience that the question will be can be done just after the 3 talks. So we are going to move to the last talk of this session. So it's a bit weird because I'm going to introduce myself as a speaker. So just let me share my screen. I'm going to talk about what is quite interesting. I'm going to talk about some project that I've done with the Public Hospital of Rennes also, and so that's why I would like to talk just after Cedric. Okay. So do you see my screen from now?
Yeah, we do.
Okay, perfect. Thank you.
We can't see your video.
You can see my video?
Now we can see.
Oh, okay. Perfect. So I would like to present you a specific project that we had with the public hospital of Rennes about using AI for pharmacovigilance. So the idea is that we are going, we had a specific project that is an ANR project. So it's public funded project, which is called InShare, for integrating and sharing health data for research. So the idea of this project is the following. There has been growing interest in conducting research with data collecting during the course of clinical care. It has been demonstrating, for instance, that electronic health records may surpass many existing registries and data repository in volume. So the reuse of this data may diminish the cost and inefficiency associated with the clinical data. So we deal here with the health big data. What is the health big data? Is the source of all the data that is collecting according to your specific, let's say, health path. So it can be produced by hospital, it can be produced in town, it can be produced by mobile device. We have a lot of IoT, health IoT now with specific connected watch, etc. And for researcher, for sure, as we have seen previously. So the sources are diverse, complex, disorganized, massive, and multimodal. So the idea here is that we want to decreasing the research cost and increasing the patient-centered research by and speeding the rate of the new medical discoveries. So we applied it specifically in this project, in one use case of this project in pharmacovigilance, that I will talk to you about that at the end. The idea of this project is that we need to share and to exploit efficiently all the heterogeneous health big data. It's coming from multi-source and at multi-scale level. So the health data is sensitive, has a limited level of interoperability, and is of very variable quality. So the idea is that it must comply with the data protection governance policies, for sure, taking into account all legal, ethical, and deontological aspects. The data are usually compartmentalized. We talked about silo previously in different information systems and are therefore, let's say, syntactically, and semantically heterogeneous. And finally, the data come from research and epidemiologic database, usually in registries or the public health agency in France, which is the information system of this public health agency is the SNIRAM, or other public institution like INSEE for the statistics institutes. And the data yield during healthcare do not comply with the same quality level usually. So the objective of our project is to use big data and artificial intelligence as disruptive technologies for this project. So we want to explore newest technology that could overcome all such issues presented previously, and we want to design an experimental proof of concept that can be helpful for conducting and achieving real-world scenarios of the secondary use of data in the field of medical research. So what we need governance models for data sharing, and we need also efficient treatment of the data using artificial intelligence, for instance. So the idea is that our approach in the project is together the stakeholders, the computer scientists, and the researcher. That is the three pillars of the big data definition, with an inherent need of scalability. So in the consortium, we had a lot of people. So we had some data provider. We have some research teams. So we have the team in a Rennes hospital, public hospital, and the LTSI laboratory, and the team which is called DMS for, let's say, large-scale health data. They provide some CDW, so it's the clinical data warehouse for the Rennes hospital, and now they have they expand this specific warehouse to a consortium of hospital. And they are interested in semantic interoperability and health data governance. We also have two different research laboratories from IMT Atlantic, which is my university, One is from data security, cryptography, watermarking, and anonymization, the identification of data, medical record and indexing, which is located in Brest in Latin. And the other one is located in Rennes, so in another town in France. It's ERISA, it's my main laboratory, on which we are experts in data streaming model, in AI mechanism, NoSQL technologies, etc., and machine learning prediction and statistical and probability modeling in order to predict and infer some new discovery on this specific data. And last but not least, we are also the French School of Public Health, so the EHESP, and the specific team from epidemiologists and biostatisticians inside this specific laboratory. So this is the research team that was involved in these consortiums, and we have we were quite lucky because we had a lot of data providers in these consortiums. So we have the agency of the biomedical, which have a specific registry, so a quite exhaustive database on the key research. We have also the cancer registry from the hospital from Poitiers. We have the center of the data center. Clinical data center from the Hospital of Brest. We have also the clinical data center for sure from the Hospital of Rennes. And we have also, it's quite rare in France, we had access to the SNIRAM, which is the national information systems that cross regime of health insurance. So the idea is that in the SNIRAM, you have all the health insurance refund to the population. So we have 1.2 billions of treatments from handled and anonymized each year to feed this NIRAM warehouse. So it was quite huge and very interesting to cross the in-town data that is available in the SNIRAM and all the clinical data that is, let's say, smaller but more exhaustive. And finally, we have also a last register on malformations of birth defect registry of Britain. So all these consortiums, for all these consortiums, we create a specific warehouse, let's say, framework on which we have worked. So from this framework, we will have a data repository and hosting. So we will crowd all the registries and the data warehouse that exist at the externally and that give us access to this health data warehouse, and then we make a copy in order to be able to work on the data directly and not working on the production data for sure. So we will have some data repository and hosting, and then we have had a specific let's say application for health and research integration. So we had semantic services, data pre-processing, aggregation, computing, and data quality and integrity monitoring also. And then, as I said, we are interested in data governance and study management. We are integrating also in the multi-source query builder and data visualization from all these data repository. And for that, we have had ID linking services in order to link the data from all this repository to the same and to try to recreate the same patient data. Of course, we had to add some data protection for all this very sensitive framework, and all the data was located only in the hospital and never be accessible from outside of the hospital. So the idea is that Inshare is kind of a trust third-party platform. We take advantage of all the best of the data warehouse and private cloud computing technologies, and having an acceptable level of data protection and sharing governance to host multi-sources health data for research project. So one example here, for example, is the kidney failure and cancers. So we want to make the link between the registry of of kidney registry and the cancer registry. And from the user point of view, the interaction with the platform consists in submitting a request for a study to the platform. So an external researcher can ask for a new project and say that, okay, I need to have this kind of information, this kind of data, and I want to study the interaction between this data, for example. So a study corresponds to one. We propose some scenarios. So this is one scenario, for example, here, which is a cross-domain study, and we can use it, for example, also for registry data and enrichment. So here in the ad hoc study from kidney and cancer, we are going to extract specific data, providing watermark data set in order to securize the data that is going outside of the warehouse. And then we are going to, after the study, to have criteria and variable selection according to a data user agreement, for instance. What is interesting here, and that's why we talk about pharmacovigilance here, is because the registry can be fed from multi-data source. So for example, we can collect comorbidities and drugs exposure in the ESRD patients. So again, we provide all these data sets as watermarks in order to protect the data that is going outside of the data warehouse. And we can get a list of patient ID and from which we can have information. So what we have done here in this specific example is that we have very nice results about the unknown side effects of the medicines interactions. So we analyze all the possibility using the pattern where two different medicines has been given at the same time for a lot of patients. And when we discover a specific pathology of this patient that happens after the medicine prescriptions, and that was not known in the specific registry of side effect and interaction of medicines, then we rate some alerts to the stakeholders, to the practitioners, to the specialists that can maybe take the decision that this is effectively a new side effect that has not been detected previously. So we use some random forest and machine learning algorithm in order to do this analysis. So the scientific agenda of this specific project has been for 5 years previously, so from 2016 to today. And so the idea of the project is the governance, data privacy and sharing procedures that have been done. We have done the data integration and the platform realization. So to describe the use case and user needs, to define data sharing governance and secure access to the platform, to define in-share platform requirements specification, PRS, and then to prepare and provide the data sets and data sources. So what is interesting is that in parallel, and this will be the conclusion of my talks, in parallel, Cedric Villani's study, the Villani reports, a study on artificial intelligence that had been published in March 2018. The President of the Republic of France affirmed his desire to make health one of the priority sectors for the development of artificial intelligence in France. And so 1 year after, so 2 years ago, in July 2019, a specific law has been raised on the organization and transformation of the healthcare system. And so there has been the creation of the Health Data Hub in France, which is a public structure whose objective is to enable project coordinator to easily access non-nominative data hosted on a secure platform in compliance with the regulation and the citizens' rights. So finally, the Health Data Hub is the scalable and nationwide version of the in-share big data, and the head of the in-share project has been one of the experts to develop the Health Data Hub, which is Marc Kudja in the Rand Hospital. So they will be able in this Health Data Hub to cross-reference and analyze the data in order to improve the quality of care and the patient support. So that's it. That concludes my talk. I hope it can raise some interest and some nice questions. So now I will stop sharing my screen, and we are going to go back in the Q&A session if you're interested in. So please do not hesitate to ask questions to our three speakers. So Platosh, Cedric, and myself. You will understand that I won't participate for the Q&A, for the Q session, and only today for this specific subsession. So please, the floor is yours. Do not hesitate to activate your microphone.
Since no one is asking question, let me begin. Mike, am I audible, Ian? So
Yes,
my
yes.
question is to Pratosh. Just as a clarification, so when you talked about this projection of the target sample via a optimization technique, are you using a trained machine learning system in the process, or you are maintaining the training data? I mean,
yeah. So we are not maintaining the training data at all. So this is
that's what I expected.
Yes. So because that would be a lot of latency. So what we do is we actually store a generative model that is built on the source data, right? But we don't save the data at all. Yeah.
Okay. And this optimization process more or less can run very quickly. It doesn't take much time.
Yeah, I mean, it depends on what is much time. It's the order of milliseconds. That's what it happens.
Yeah, that's what I was expecting. Thank you. It's a very, very interesting idea.
Thank you. Thanks.
Thank you.
If there is another question. So maybe I will ask the same question for the two other speakers of this session. So I will start with Cedric. If you imagine that you have infinite funding, what is in the next 5 years for you the next most important problem that has been taken into account and resolved in the AI for healthcare, in your point of view, and in the point of your research?
I'm more emergency physician than a researcher, but to my point of view, the clinical point of view, I would like to construct systems that can detect critical events for the patient, such as strokes or heart attack or cardiac arrest or sepsis. And I think that could be maybe the from a practical point of view, the main objective of the studies to be performed in the next 5 years, because I think it would be very useful for patients. And to do that, you have to increase the interoperability between databases and data warehouse, just like your presentation was about. We cannot hear you, Jan.
Sorry, I shut down the microphone. Thank you very much, Cedric, for this
Thank you.
Interesting perspective. So same question for Pratosh.
Yeah. So I would say that, if you can solve some of these ML problems using, say, 1% or even 5% of the data that we use today, so that would be a great success. So you should not scout for millions of X-rays annotated. If you can just do it with 50 or 100 samples, can solve a lot more problems.
Okay, thank you very much. Is there some other question from the floor? No other question?
Ian, I have a small question for you. So this consortium that you have, I mean, so do you allow like people from outside the consortium, not outside the country, I'm saying within France, to access the data if they want? And second question is that you mentioned about the patient ID. I believe that's a unique patient ID which can be used by every hospital or something like that. So hospital and things like that.
Yeah, so in France, the last 2 years there have been a lot of moving. So maybe Cedric can say a bit about that also. But the idea of this consortium, so the InShare consortium, was a proof of concept that give rise to the creation of the Health Data Hub. So now the Health Data Hub is national-wide, and so the idea is that any researchers in this field can ask and can provide some information, some new project on the data in order to study to make their studies. So, for example, there have been for the 2 last year there have been more than 1,600 submitted project to the Health Data Hub from the research side. So it's not limited.
You are not audible.
Oh, sorry.
You are not audible.
It's maybe my.
We cannot hear you. Now we can hear.
Yeah, okay, maybe it's my internet connection that was a bit unstable. So what I said is that in the Health Data Hub, the idea is that you can be outside of the consortium because it's a national-wide platform only funded by the government, by the French government. And the idea is that there is more than 1,600 projects that has been submitted from the last years. From the 2 last years. So the idea is that you can ask for a specific collection of data, a specific collection of patients that has specific morbidities, for example, and then using that you can provide you can get some info, some data in a secure way in order to do your analysis. And according to the ID of the patient, yes, we now be able to link either the data are a bit, let's say, de-identified. We are able to link without putting in notification of the data. So you cannot go to the specific person, so to a specific human being, let's say, but you are able to link the file and the line in the different registry in order to recreate anonymized health file, for example, and so you can then use it in terms of in-town and clinical data for the same person.
Again, there may be problem with the internet connection.
Oh, I'm sorry. I'm really sorry that.
That's it happens. It happens. Now it is okay again.
Okay. So if there is some other remark, question, general discussion from all the panelists, from all the session of this morning, is there someone who wants to take the microphone and give a word? Do not hesitate to just unmute your microphone to give your insight. Okay, so if there is no more specific comments or specific questions, I think we are going to close this first day session workshop. So again, I want to really thank warmly all the people that has been presenting this morning, and all the valuable discussion that we had this morning during this first day of the workshop AI for Healthcare in the framework of the Knowledge Summit 3. So tomorrow we will have some very interesting talks also, and with very interesting discussion with a lot of other colleagues from India and France. So do not hesitate to join us tomorrow morning directly in this room at 9:30. And in the meanwhile, I wish you a very nice evening in India and a very nice afternoon in France, and hope to see you in 9:30 in France morning and 2 p.m. in India for the second day of this workshop. Thank you very much to be there. Thank you very much, Nikhil, to be here with me to co-animate this workshop.
Thank you, Ian, for taking the lead and conducting the workshop in an excellent manner. Thank you very much. And see you everybody tomorrow morning. No, tomorrow afternoon in India and morning, of course, in France.
Exactly. Thank you. Thank you very much.
Thank you. Bye-bye.
Thank you, everyone. Bye-bye.
Bye.
Thank you.
Thanks, bye.